Let's get into the contents. Alcohol dehydrogenase, shortly called as ADH, catalyzes the reversible oxidation of alcohols to their corresponding aldehydes or ketones. They use NAD plus as their cofactor. In case of human ADH, it is classified into 5 major classes based on sequence homology, kinetic behavior, and tissue distribution. These isoforms vary in substrate specificity, kinetic property, and physiological roles. Coming to the class 1 ADH, we have 3 more subclasses in ADH. It is ADH 1A, 1B, and 1C. These enzymes are located in different tissues like liver, gastric mucosa, and kidney. They are very specific for ethanol, retinol, and other short chain alcohols. Coming to the importance of this enzyme, they are very important for the ethanol metabolism. Class 1 ADH is responsible for 90% of ethanol oxidation in the liver, where ethanol is converted into acetaldehyde and acetaldehyde is further converted into acetate. Since acetaldehyde is toxic and it causes flushing, nausea, and tissue damage. Genetic polymorphism and alcohol sensitivity of the variants of ADH1 show higher catalytic activity, and it is very common in Eastern Asian populations, which provides them protection against alcoholism. ADH1 has a major role in vitamin A metabolism, where it oxidizes retinol to retinal, which is a crucial conversion process for vision and epithelial differentiation. Next is your class 2 ADH, and it is distributed in liver and GI tract. Coming to the substrate specificity, it is very specific for higher alcohols, fatty alcohols, and retinol. This ADH class 2 has a greater importance in ethanol metabolism. It is active when blood levels are elevated, and it is important in chronic alcoholics. It is also involved in drug and xenobiotic metabolism, where it oxidizes aromatic and long chain alcohols and contributes to detoxification pathways. And it also contributes for liver toxicity, where the produced acetaldehyde due to heavy alcohol consumption is going to increase your oxidative stress. Class 3 ADH is also called as formaldehyde dehydrogenase or ADH5. It is distributed ubiquitously in almost all the tissues like liver, lung, brain, and kidney. Coming to the substrate specificity, it is very specific for formaldehyde, as the name suggests, and it has very low affinity for ethanol. The main clinical importance of this class of alcohol dehydrogenase is in formaldehyde detoxification, where toxic formaldehyde is converted into formate, and it protects the cells from DNA and protein damage. And it also protects the cells from cancer and oxidative stress, because formaldehyde is a carcinogen, and impact ADH5 activity is related to increased cancer risk. It is also involved in nitric oxide regulation, where it is involved in S-nitroglutathione metabolism. It influences vascular tone and immune response. Coming to the class 4 ADH, it is distributed in the upper GI tract, oral cavity, esophagus, and stomach. It is very specific for retinol and ethanol. It is a very important enzyme in drug metabolism, where the first pass metabolism of alcohol is mediated by ADH4. It metabolizes ethanol before it reaches systemic circulation and reduces blood alcohol concentration. It protects body against alcohol toxicity. It acts as a barrier enzyme in the gastrointestinal tract. Upper digestive tract cancers, which are due to increased acetaldehyde production locally and are linked to oral and esophageal cancers in alcohol consumers. Coming to the last class of alcohol dehydrogenase, it is distributed in liver. It is very specific for steroid and fatty alcohol metabolism. The actual role and the specificity is not driven detail. It is clinically important, and it has a major role in hepatic metabolism under specific condition. Coming to the overall significance of alcohol dehydrogenase, It contributes in alcoholism and genetic susceptibility, where it influences drinking behavior, risk of dependency, and severity of alcohol induced damage. It has a major role in drug metabolism to metabolize methanol to formaldehyde, ethylene glycol to glycoaldehyde, and impairment in these metabolism leads to metabolic acidosis and visual impairment due to methanol poisoning. It has a major role in liver disease, in cancer by avoiding accumulation of aldehyde, which is associated with various cancers such as esophageal, head and neck cancer. It is also used as a biomarker for liver damage and a target for alcohol aversion therapy and toxic alcohol poisoning management. These are all the different roles of alcohol dehydrogenase and their clinical significance. Let's come and learn another interesting enzyme in the next class. Thank you for